Good morning, I'm Niharika Mishra.
And I'm June Smith.
Our research was testing non-dairy fermentation of commercially used probiotics. The biggest challenge was to make a plant-based product that mimics or function like milk-based fermentation products, controlling the fermentation process to achieve a product that consumers are familiar with as far as texture, mouthfeel, and taste. Since plant-based extracts contain different sugars for fermentation, that could impact the product's functionality, shelf life, and flavor.
Why are we looking at this research? Due to lactose intolerance and dietary restrictions, some people lack the benefits of probiotic-rich fermented foods. Non-dairy food options that mimic or replace dairy foods, such as plant-based milks and yogurts, have become a growing cultural necessity as well as an important economic component of many company product portfolios. According to MarketWatch in 2019, sales of plant-based milks were up 9% to $1.6 billion and now make up 13% of total milk sales, while cow's milk sales were down 6%. Plant-based cheeses sales were up 43% to $124 million, and plant-based yogurt sales were up 55% to $162 million. People are willing to pay premium prices for this plant-based foods, and the Plant-Based Association stated that in 2019, sales were up 6% overall. It is necessary to determine if dairy-derived lactic acid bacterial cultures can effectively ferment plant-based milk substitutes. 3 plant extracts, almond, coconut, and oat, were selected and tested at 3 inoculum levels, and the pH monitored over 420 minutes at 37 degrees Celsius. The commercial strains used in this study included all of the following in this table. However, we focused on YFLO2, LGG, and casei 431. Since some of these strains are mixtures, we were left with the challenge of not knowing the exact bacterial composition. The concentration of each strain may vary in the mixture. We wanted to use currently used commercial mixtures to determine if these could be substituted into non-dairy beverages and have similar acidification and fermentation results.
MRS broth and agar were used to propagate most lactic acid bacteria in this study. For MRS cysteine, 5% L-cysteine hydrochloride solution was used for propagation of Bifidobacterium strains. M17 broth and agar were used to propagate Streptococcus thermophilus strains. Bromocresol purple galactose agar was used for differentiation of Streptococcus thermophilus from Lactobacillus bulgaricus in mixed starter cultures.
To test the ability of the probiotics to survive in non-dairy beverages, we inoculated into each beverage and incubated for 5 hours. Surviving organisms were quantified on MRS. casei 431, LGG, and YFLO2 were detectable in quantities similar to the inoculated concentration following 5 hours of incubation. Typically, milk ferments milk sugar lactose to lactic acid and lowers the pH of the product. Since plant-based extracts lack the lactose, other sugars must be used for fermentation. Fermentation capabilities of each individual lactic acid bacterial strain were assayed with the API 50 carbohydrate panels.
All bacterial strains fermented galactose, glucose, fructose, mannose, esculin, and tagatose. Since the most common sugars available for fermentation in plant-based extracts include the disaccharides, sucrose and maltose, and the monosaccharides, fructose, mannose, and glucose, bacterial strains tested for this research appear to have the capacity to utilize common sugars found in plant-based extracts. Our experimental design to determine the fermentation capacity of the lactic acid bacteria in plant-based extracts included the following steps. Each strain was isolated and grown anaerobically at 37 degrees Celsius in MRS broth or MRS cysteine broth and plated for viability. Initial trials tested the ability for acid production of each individual strain in 3 plant-based extracts: almond extract, coconut extract, and oat extract.
Secondary trials tested the optimal concentration by volume of individual strains to reach the desired pH range, 0.5%, 1%, and 2% volume per 99 mL of extract. These samples were incubated in a water bath at 40 degrees Celsius in triplicate, and from this data, it was decided to continue further trials with the oat extract and selected isolates based on acidification within 4 hours. Trials continue with the use of selected combinations of probiotics. Inoculum of either 0.5% or 1% volume by volume of frozen pellets, single, double, and triple strains were combined. KCI 431, KCI 431 plus LGG, and KCI 431 plus LGG plus YFLO2. They were incubated in a water bath at 40 degrees Celsius in triplicate. Readings were taken at time 0, 60, 120, 180 minutes until pH 5 was reached. Then samples were plated on MRS and incubated at 37 degrees Celsius anaerobically for 48 hours and enumerated.
Extract. All the plant extracts, oat extract performed the best. All the test cultures, both plants and single strains, decreased the pH of the oat extract over the test period. At 4 hours, the pH decreased from pH 7.70 to pH 6 for all cultures except for KCI 431, while at 1% inoculum concentration, YFLO1 decreased the pH to 5. At 2.0% inoculum, all the cultures were able to decrease the pH to 5.5. At 420 minutes, most cultures could decrease the pH below 5.5.
Oat extract. As we can see on this graph, the oat extract performed the best and decreased the pH to 5 in about 300 minutes with 0.5% and 1% inoculum. Due to the success with the oat extract, it may be possible to use selected single lactic acid bacterial strains to achieve an acceptable commercial fermentation in 4 hours.
Experimentation is to create a stability model of 2 probiotics, BB12 and LA5, and we used oat milk as the plant extract. We concentrated the colonies by centrifugating them for 15 minutes, and then we used 1% of probiotics to inoculate the plant extract. The trial ran for 14 days. We enumerated them at day 0, day 7, and day 14.
From the data, we can understand that BB12 survived best at 5.5 pH. One thing we derived about LA5 was they didn't survive in oat milk, and we're trying to understand why. There may be some industrial oat milk ingredient that may be inversely impact LA5 survivability.
The following 4 oat milk products have some sort of phosphate compound as stabilizer, but Simple Truth does not have dipotassium phosphate, which is going to be our experimental basis moving forward. We will test Simple Truth to see if it increases the survivability of probiotics. Utilizing the 4 different brands of oat milk we inoculated commercially used probiotics with 7 different strains. Our motive was to see which oat milk suits best for probiotics, on the basis of which we can decide which ingredient inhibits or promotes the growth of probiotics. Our findings suggest that dipotassium phosphate is acting as an inhibitor to the growth of these commercially used dairy probiotics. Simple Truth gave the best results of probiotics except for KCI 431 and BLC 48. LH32 showed growth in Simple Truth. BLC 48 grew best in Planet Oat. KCI 431 grew only in Chobani and Oatly. Continued research will be testing the survivability of probiotics in the oat milk brands that does not have phosphate stabilizers in it, so that we can derive a direct relation.
Research, our data showed that oat extract inoculated with most lactic acid bacterial strains yield the greatest pH drop in 4 hours. Coconut extract and almond extract showed much less decrease in pH with little difference between the two. We evaluated the survival and stability of LA5 and BB12. We would like to thank the Microbiology Department at Weber State University, Karen Mann, Dr. Michele Culumber, Dr. Craig Oberg, and the Weber State University Office of Undergraduate Research and Chobani Incorporated.